All right. Thanks for coming back to us on the New Equity Network show. We are on the show here with some really earth-shattering technology. Roger White, President and CEO, Theralase Technology. Symbol on the TLT, TSXV, and on the OTC pink sheets, TLTFF. Website, Theralase.com. All right, Roger, you are working on cancer treatments, too, and some anti-cancer technology. And my co-host, Ed Baxter, has got some questions for you. Let me let you jump in there, Ed, and talk to Roger for a sec.
Good morning, Roger. First of all, I'd like to thank you for coming on and giving us a chance to hear about this great company and great technology.
My pleasure, Ed.
A couple of things kind of stand out as my background's in the securities world, the investment banking world. So a couple of things stand out when I look at and hear the story, and I kind of like to have address a couple of things here. We've seen a number of different technologies, both cold and warm, with regard to lasers, and one particular company I know of does a similar situation where they're actually targeting cancer cells within the body deeply and actually freezing them and allowing them to actually be taken out of the body also. Can you compare and contrast the different or some of the different technologies associated with the cold laser and maybe the pinpointing or freezing of malignant cells within the body, and how the different techniques work depending on what cancers are being attacked?
Absolutely. I think that the three main treatment methodologies for cancer involve surgery, where you try to excise the cancerous tissue out. You're looking at chemotherapy, where you try to use very powerful cytotoxic or cell-killing drugs in order to try to destroy it. Or you use radiation, where you use linear accelerators to try to destroy the vasculature for it. So if you're using cold therapy or hot therapy, it's very similar to a surgical application, because you're trying to either use cryotherapy, where you're trying to freeze it to destroy those cells, and then you're going to remove them afterwards. Or there is technology where you use heating, or you use radio frequency, or you use X-rays, or you do different things that are going to produce heat, and again, try to destroy it that way. Our technology is, I think, significantly different, because here we're, both our divisions, the therapeutic division and the cancer division, we're operating on a cellular basis. So here, we're getting down to the molecular or the cellular level of the tissue. So in the healing, we're actually dealing with the mitochondria of the cell, so to accelerate the production of adenosine triphosphate, which is a natural food source, in order to heal that cell. In the cancer technology, our drug is what's known as a small molecule. So it's a very, very small molecule. It's able to dissolve completely into water. And in that case, we can have it cross the cellular membrane, and it goes in and it grabs onto different organelles within that cell. It's benign until we light activate it. Once we light activate it, it produces a very reactive form of oxygen known as ROS, or reactive oxygen species, which destroys the cell from the inside out. And it destroys only the cancer cells. So versus trying to do a kind of a shotgun approach, so to speak, with cryotherapy or heat therapy, this is much more of a rifle approach.
Well, being an East Texan, you're speaking my language on the firearms thing. So, thank you for clearing that up for us. So essentially, it's a two-step process where you're inviting in the drug itself, and then you're activating the drug on a very specific and laser-like basis, if you'll pardon the pun there.
Absolutely.
Okay. Obviously, you're moving toward your clinical trials with regard to the different bladder projects and other things that you have going as far as cancer is concerned. Kind of give us, if you would, please, a little bit of a timeline of how you expect to commence that program, and when you expect to have reasonable information about what the efficacy of it is.
Well, in the drug approval process, in Canada, it's known as a clinical trial application. In the U.S., it's known as an investigational new drug application, or IND. There's really a number of steps that you need to have in place. You need to be able to manufacture the drug to a GMP, or good manufacturing practice standards, meaning that it has a certain purity, has a certain yield, that you can use it in human applications. Second thing is you need to have toxicology analysis completed, so you need to know what the minimum effective dose is, what's the maximum tolerated dose. So really finding the goalposts, so to speak, as to what's the low point and what's the high point for your drug for entering it into the human body. And the third piece is really a detailed clinical plan as to how you're going to treat a disease like non-muscle invasive bladder cancer or any type of cancer for that matter. So Theralase is currently working on all three of those and plans to have that completed in third quarter of this year. So it would be in the July to September timeframe for us. Having submitted all that to Health Canada, we'd be in a position to start enrolling patients for non-muscle invasive bladder cancer in fourth quarter of this year.
Fabulous, fabulous.
All right. When you're looking to enroll patients, you said fourth quarter of this year, right?
Mm-hmm.
All right. Give us an idea where you're going to start on what applications you're going to start with, and where you think you're going to end up.
Well, the process is going to start in Canada with Health Canada, because we've met with Health Canada. We've reviewed the plan as to what we want to accomplish. They seem to be on board with that. Of course, we'd need their final approval to start. In and around the summer, we'll be meeting with the FDA, and we'll be talking to them about the plan and about the enrollment strategy, et cetera. And with their blessing, we'd be able to start enrolling patients on the U.S. side of the border. The start is really non-muscle invasive bladder cancer. So with that, it's a very deadly disease. I think it's the fifth most common in the world. I think there's about 75,000 Americans that are inflicted with it each year, and about 15,000 deaths. So it's quite a traumatic condition. So what we're doing here is we're actually looking to instill our drug into the bladder intravesically. So we'll be able to, bladder cancer is a cancer that grows from the inside out. So by attacking it from the inside of the bladder and moving out, we're going to be able to have our drug localize into those bladder cancer cells, and then when we light activate it, we'll be able to destroy it again from the inside out. So we're really looking forward to proving the safety and tolerability of our drug, and also the efficacy in and around the fourth quarter of this year.
Okay. Ed's got one more for you.
Well, you brought up an interesting concept when you just mentioned that we've done some deals for different type of clinical trial companies and other drug companies, and one thing that kind of stands out, and if you kind of elaborate on just a tad bit, is that kind of the parallel universe you're walking right now with regard to an invasive drug versus a non-invasive light therapy to activate that drug. Can you kind of shed some light on that, if you'll pardon the pun, on how that parallel universe works in the research world?
Absolutely. With a systemic drug or an oral drug, something that you're going to be taking on a daily basis, there's always risk for toxicity, because your body's very efficient. Your renal system, your liver, your kidneys is very efficient of taking that substance or foreign substance out of the bloodstream, and it's going to reside into the liver, the kidney, etc. And there's a lot of risk for toxicity. So there's a lot of drugs out there that can cause a lot of contraindications, a lot of side effects associated with it. Our methodology is quite different. With bladder cancer, since the cancer starts from the inside of the organ and grows out into the organ, what we're doing is we're planning to instill the drug, because it's a water-soluble drug, into the bladder through the urethra, have it situate there for about an hour, localize or be absorbed into those bladder cancer cells. Then you'd void the bladder. We would fill the bladder again with water to give it shape, and we would use what's called a fiber optic bundle. So we would insert fiber optics, which look like fishing line, which light up, inside the bladder, activate the drug, destroy all the cancer cells, and you basically urinate it out. Whole procedure looks like it could be done in about 3 hours, without any of the side effects associated with a lot of these systemic pharmaceutical drugs.
All right. Ed, do you have one more question? He's waving at me madly.
Yeah.
He's gotta get there.
The one thing that stands out there, too, is I think about companies like Amgen and a few other the big drug manufacturers, and Epogen and some of those things. And and I know what the toxicity level of some of those drugs are. Obviously, if your trial goes as expected, which you think it'll be expected, is it possible you might make an enemy or maybe a friend of one of those companies?
Well, I think that technology is always developing. So I think that if you can provide something which can be used adjunctively, which can be used in combination, for example, with a pharmaceutical, or it could be used alternatively for patients who don't respond. Like the standard of care right now for bladder cancer is what's called BCG, Bacillus calmette-guerin, where they use kind of a bacteria in order to slow down or destroy the disease. there's what's called a refractory population. So 25% of the patients who get that gold standard treatment, it doesn't work for them. So that's our initial population, is for the patients that have had the therapy and it doesn't work for them, then those are ones we're trying first. So I think that there's lots of opportunity to partner with Big Pharma for applications where their drugs are refractory or not working on certain populations.
All right. Roger, we've got to call an end to the interview, but I want to thank you so much for coming on to the show. I really appreciate your time.
I appreciate the opportunity.
Thank you. All right. Roger White, President, CEO, Theralase Technology, TSXV on the TLT, and TLTFF on the OTC pink sheets. It is Theralase.com. We'll be back on the other side of this break with Andrew Whalen, CEO, Bioelectronics Corporation.